Hello friends and welcome. Today we are going to discuss an important topic, that is clinical biochemistry, basic concepts and scope. The main objectives of today's lecture are: first, to enable the students to understand the basic concept of clinical biochemistry. Number second will be to discuss different biochemical tests. Number third, to give a brief idea about biochemical investigations of blood specimens. And number fourth, to give the students basic idea about scope of clinical biochemistry. So let's first discuss the basic concept of clinical biochemistry. Clinical biochemistry is defined as the special branch of medicine dealing with measurement and interpretation of the physicochemical conditions and dynamics in healthy and diseased humans, thus contributing to our pathophysiological understanding and thereby to diagnosis, therapy, prognostication and research of disease. It's also defined as branch of medicine concerned with developing and carrying out clinical analysis of body fluids and other biological materials for the diagnosis and therapy of disease. Clinical biochemistry is an important applied subdiscipline of medical biochemistry, also known under the names of clinical chemistry, pathological biochemistry or chemical pathology. Clinical biochemistry is concerned with methodology and interpretation of biochemical tests performed on body fluids and tissues to support diagnosis, treatment and monitoring of the disease. Clinical biochemistry is one of the most rapidly advanced areas of laboratory and clinical medicine. The marked increase in the number and availability of laboratory diagnostic procedures has helped in the solution of clinical problems. Biochemical tests are crucial to modern medicine. Most biochemical tests are carried out on blood using plasma or serum, but urine, cerebrospinal fluids, feces, kidney stones, pleural fluids, etc. are sometimes required. Plasma is obtained by collecting blood into an anticoagulant and separating the fluid. Plasma phase from the blood cells by centrifugation. Serum is corresponding fluid phase when blood is allowed to clot for many, but not all, biochemical tests on blood. It makes little difference whether plasma or serum is used. Clinical biochemistry tests. The biochemical investigations on blood, plasma, serum carried out the clinical biochemistry laboratory may be grouped into different types. And number first is discretionary or on-off tests. Most common clinical biochemistry tests that are designed to answer specific questions. For example, does the patient have increased blood, urea, glucose concentration? Normally, these tests are useful to support the diagnosis. Number second is biochemical profiles. These tests are based on the fact that more useful information on the patient's disease states can be obtained by analyzing some more constituents rather than one. For example, plasma electrolytes, which contains sodium, potassium, chlorine, bicarbonate, urea. Liver function tests, serum and bilirubin, ALT and AST. Number second is dynamic function tests. These tests are designed to measure the body's response to external stimulus. For example, oral glucose tolerance tests to assess the glucose homeostasis. And number fourth is screening tests. These tests are commonly employed to identify the inborn errors of metabolism and to check the entry of toxic agents, for example, pesticides, lead, mercury into the body. Metabolic workup tests. The programmed intensive investigations carried out to identify the endocrinological disorders come under this category. The term emergency test is frequently used in the clinical laboratory. It refers to the tests to be performed immediately to help the clinician for proper treatment of the patient, for example, blood glucose, urea, serum, electrolytes. There are over 400 different tests which may be carried out in clinical biochemistry laboratories. They vary from the very simple, such as the measurement of sodium, to the highly complex, such as DNA analysis, screening from drugs, or differentiation of lipoprotein variants. Many high-volume tests are done on large automated machines. Less frequently performed tests may be conveniently carried out by using commercial prepared reagents packaged in kit form. Some analyses are carried out manually. Now let us discuss the specimen collection. The biological fluids employed in the clinical biochemistry laboratory include blood, urine, saliva, septum, feces, tissues and cells, cerebrospinal fluids, peritoneal fluids, stones, etc. Among these, blood, directly or in the form of plasma or serum, is frequently used for the investigations in the clinical biochemistry laboratory. Now let us discuss how blood is collected. Venous blood is mostly commonly used for the majority of biochemical investigations. It can be drawn from any prominent vein, usually from the vein on the front of the elbow. Capillary blood obtained from a finger or thumb is less frequently employed. And arterial blood, usually drawn under local anesthesia, is used for blood gas determinations. Let us discuss some of the precautions for the blood collection. Number first is use of sterile needles and syringes, cleaning of patient's skin, blood collection in clean and dry vials, tubes are some of the important precautions. Now, let us discuss biochemical investigations of blood specimens. Biochemical investigations can be performed on 4 types of blood specimens, that is whole blood, plasma, serum and RBCs or red blood cells. The selection of the specimen depends on the parameter to be estimated. Whole blood, usually mixed with an anticoagulant, is used for the estimation of hemoglobin, carboxyhemoglobin, pH, glucose, urea, non-protein nitrogen, pyruvate, lactase, ammonia, etc. But we have to note that for glucose determination, plasma is preferred in recent years. Plasma obtained by centrifugation, the whole blood collected with an anticoagulant, is employed for the parameters like fibrinogen, glucose, bicarbonate, chloride, ascorbic acid, etc. Serum is the supernatant fluid that can be collected after centrifugation of the clotted blood. It is the most frequently used specimen in the clinical biochemistry laboratory. The parameters estimated in serum include proteins that include albumin, globulins, bilirubin, cholesterol, uric acid, electrolytes, enzymes like ALT, AST, LDH, CK, ALP, ACP, amylase lipase and vitamins. Red blood cells are employed for the determination of abnormal hemoglobins, glucose 6-phosphate dehydrogenase, pyruvate kinase, etc. Now let us discuss commonly performed clinical tests. Number first is liver function test, in which we count total bilirubin. Increased in excessive RBC breakdown such as hemolytic anemia or impaired liver function or some sort of obstruction such as tumor or gallstone. Liver enzyme levels increase following damage to the liver tissues. Number first is alkaline phosphate, greatly increased in liver tumors and lesions and moderately increased in disease such as hepatitis. Alanine aminotransferase, formerly called as SGPT, increases up to 10x in cirrhosis, infections or tumors and make up to 100x in viral or toxic hepatitis. Aspartate aminotransferase, formerly called SGOT, increased in liver disease but also in heart attacks. Gamma glutamyl transferase, often used to monitor patients recovering from hepatitis and cirrhosis. Lactase dehydrogenase, increased in liver diseases and following heart attacks. Cardiac function tests. Creatine kinase, widely used to diagnose and monitor heart attacks. Troponins, only present in heart muscles, making it a more accurate indicator of heart attack than creatine kinase. Now, we will discuss thyroid function tests, and in which we will discuss thyroid stimulating hormone. Inverse relationship to thyroid function, the higher the TSH, the lower the thyroid function and vice versa. Other less common thyroid tests include T3 and T4. Hypothyroidism, underactive thyroid gland, and hyperthyroidism, overactive thyroid gland. Now, let's shift to the scope of clinical biochemistry. Clinical biochemistry has a very wide scope in all walks of life. In order to study the scope of clinical biochemistry, we must have the understanding of the following facts. The study of clinical biochemistry helps us to know how the enzymes accomplish their catalytic tasks. Hundreds of pure enzymes each more or less specific for one chemical reactions, have been isolated and many crystallized. The study of clinical biochemistry helps us to know the synthesis of hundreds of new molecular species, intracellular accumulation of inorganic ions and organic compounds against concentration gradients, and the performance of the mechanical work. The rapid development of clinical biochemistry in the recent years has helped scientists involving some of the most challenging and fundamental problems in the biology and medicine. The scope of clinical biochemistry can be seen from the fact that it is used in many daily activities. It is used in clinical diagnosis, manufacture of various biological products, treatment of disease, in nutrition, etc. The study of clinical biochemistry helps one understand the actual chemical concepts of biology. This is concerned about the internal chemistry of biological systems of the animals as well as plants. Since it is so vast and the new phenomena are discovered every year, it is taught as a separate branch of biology. One can study clinical biochemistry as a part of graduation or post-graduation. So let's see the importance of clinical biochemistry in medicine. Clinical biochemistry is a valuable subject in medicine without which there would have been no such advancement in the field. Physiology. Clinical biochemistry helps one understand the biochemical changes and the related physiological alterations in the body. Pathology of any disease is studied through the biochemical changes. If we now look at the pathology, based on the symptoms described by the patient, the physician can get a clue on the biochemical change and the associated disorder. For example, if a patient complains about the stiffness in the small joints, then the physician may predict it to be the gout and get confirmed by evaluating uric acid levels in the blood. So, nutrition deficiency. In the present scenario, many people rely on taking multivitamins and minerals for better health. The function and role of the vitamins in the body are described only by biochemistry. So, hormonal deficiency. There are many disorders due to hormonal imbalance, especially in women and children. The formation, role of hormones in the normal body function is taught in biochemistry, by which the physician can understand the concerned problem during treatment. So, let's discuss importance of clinical biochemistry in nursing. In nursing, importance of clinical biochemistry is invaluable. When a patient is in the hospital, nurses need to keep a watch on how his condition is progressing through clinical biochemistry. Almost all the diseases or disorders have some biochemical involvement. Some of the diagnosis of any clinical condition is easily possible by biochemical estimations, and that may involve kidney functioning test. For example, in kidney disorders, other chemotherapy treatments, etc., urine test helps understand the extent of excretion of drugs or other metabolites, the change in pH, the color of the urine, etc. While as blood tests, in diabetes biochemical analytics test for blood glucose levels above 150 milligrams per deciliter helps one understand the severity of the diabetes disorder. Another biochemical test for ketone bodies in the urine also indicates the stage of diabetes. The appearance of ketone bodies or ketonuria is mostly the last stage of diabetes. Now the liver function tests. Liver function tests helps to understand the type of disease or damage to the liver, the effects of any medication on the liver, etc. Serum cholesterol tests, that evaluation of blood cholesterol levels and other lipoproteins helps understand the proneness of the patient to the cardiovascular disease. Now let us discuss importance of clinical biochemistry in nutrition. In nutrition, biochemistry describes the food chemistry for maintenance of health. Optimum intake of many biochemicals like micronutrients, vitamins, minerals, essential fatty acids and water is necessary. Food chemistry gives an idea of what we eat, that is, its components like carbohydrates, proteins, fats, etc., and also the possible physiological alteration due to their deficiency. The role of nutrients. Due to the biochemistry, the importance of vitamins, minerals, essential fatty acids, their contribution to the health were known. Hence, there is a frequent recommendation for inclusion of essential amino acids cod liver oil, salmon fish oil, etc. by physicians and other health and the fitness experts. The nutrition value of the food materials can also be determined by biochemical tests. So, let us shift our focus to the importance of clinical biochemistry in pharmacy. In a pharmacy, many drugs are stored for regular dispensing. Drug constitution. Biochemistry gives an idea of the constitution of the drugs, its changes of degradation with varying temperature, etc. How modification in the medical chemistry helps improving efficiency, minimize side effects, etc. And the half-life, that is a test done on biochemical drugs to know how long a drug is stable when it's kept at so. And it also helps in drug storage. The storage condition required can be estimated by the biochemical tests. For example, many enzymes, hormones are stored for dispensing. These get deteriorated over time due to temperature or oxidation, contamination, and also due to improper storage. Now the drug metabolism. It also gives an idea of how drug molecules are metabolized by many biochemical reactions in presence of enzymes. Now let us discuss some of the biochemical tests. These tests helps to fix specific half-life or date of expiry of drugs. Let's shift our focus to importance of biochemistry in the plants. Biochemistry of plants gave a way to the breakthrough of how food is synthesized in them and the reason why they are autotrophs, that is, not dependent on other living beings for food. Biochemistry in plant describes first the photosynthesis. This describes how carbohydrates are synthesized by the use of sunlight, carbon dioxide and water in the green leaves of a plant. It also goes on to explain about different complex enzymes involved in the process to combine the energy of sun within the molecules of water and carbon dioxide in the formation of carbohydrates. Biochemistry in plant describes the process of respiration. Biochemistry in plants describes the different sugars. Biochemistry defines different types of carbohydrates formed in the plant like trioses, pentaoses, hexaoses, heptalosis. Heptalosis are the carbohydrates which go on to form the nucleic acid, that is deoxyribonucleic acid, actually DNA, and ribonucleic acid, that is RNA. Biochemistry in plants describes plants' secondary metabolites. Clinical biochemistry also describes how the plant products like gum, tannins, alkaloids, resins, enzymes, phytohormones are formed inside the plant. Other functions, it also describes how plants' fruits get ripened, how plant seeds germinate, the respiration process inside the plant cell, how proteins and amino acids are formed on rough endoplasmic reticulum and fats are formed on smooth endoplasmic reticulum. That's all we have in today's deliberation. Hope you understand it well. Goodbye.